Hi everyone, and welcome to this training session. Today we are going to dive into session 2.4, protein engineering approaches. As new members of the iGEM team, you find sometimes you need to tweak the DNA that you find in nature. In this tutorial, we're going to cover the core PCR-based methods that we use to rewrite the genetic code and modify the proteins for our experiments. So what exactly is protein engineering? It's the process of developing useful protein by deliberately alter the amino acids sequence. So we do this to change the protein's function and possibly improve their stability or function, or even change where it connects inside the cell. So to make these changes, we rely on the PCR reaction. So while you probably learned that PCR could be as a copy machine of DNA in our lab, we treat it more like a text editor. So it could be our primary tool for inserting, deleting, or mutating specific parts of the gene instead of reorder it again. So let's look at our first major technique, which is called the overlap extension PCR. This allows us to insert the codes or delete the unwanted ones. So if you look at the diagram, you'll see how we use the two round processes. But to be noted, over on this paragraph, on this picture, you see that it only says two round PCR. The final product actually is formed in the third round of the PCR reaction. First, let's look at the insertion. Imagine we want to insert a DNA fragment into our gene, no matter it's a single base pair or it's a few base pair long. In the first round, we amplify these two DNA fragments separately with specific primers that contains a bit of overhang for another DNA fragment. So this trick is that after the first round of replication, these original DNA and DNA to be inserted will contain a small tail that matches another DNA fragment. In round two, that these matching tails will find each other and anneal. This which creates a bridge that allows the polymerase to stitch the red insert seamlessly with the purple one. As the product of the second round PCR, if you continue amplifying the product you gain, you have your new DNA fragments that fusioned your DNA to be inserted into the original DNA. This is how PCR allows you to insert the DNA sequence that you want into your designated original sequence. Now let's look at the right deletion. So here we want to remove a domain that could possibly create harm or decrease the efficiency of our DNA fragment. First of all, we design primers that flank the section we want to cut out. So here is the primer that matches the end of the section we want to cut out, and another primer is on another end. Crucially, the primer on the left fragment needs to have a tail that matches the right fragment. So during round two after replication, the middle section is skipped entirely as the tail in these primers will stitch together. So we got our targeting DNA sequence after deletion and send it for further amplification. Sometimes we don't want to add or remove a huge chunk or huge amount, a part of our DNA sequence. We just want to change one single letter, which means one single base pair mutation. You can call it single nucleotide polymerase, single nucleotide polymorphism, or SNP. This could be done by PCR or either Gibson assembly. First, let's look at site-directed mutagenesis by PCR. This is only targeting a small fragment of your gene of interest instead of mutagenesis on a whole plasmid. By specifically design your PCR primers, you can realize insertions of single nucleotides, substitutions of a nucleotides, or deletion of a nucleotides on your designated fragments. And after such mutagenesis, you can assemble these fragments onto your backbone as usual, and you can get your altered PCR, get your mutated DNA product from your host cell. You can use this method when you did not assemble your plasmid yet, or when you just obtained your ordered DNA. Also, you can perform a direct site-directed mutagenesis by using Gibson reaction. This could be used when your gene of interest has already assembled into a plasmid, or you're directed mutating it from the stock. First, we use the inverse PCR, which already has a mutation on the tail of our interested mutation point. And after the PCR, the plasmid will be opened for a linear straight line DNA. And you can see the mutation is at the end of this linear DNA sequence. Once we have the linear DNA, we still obtain the overhang in both ends of this linear DNA sequence. Afterwards, we can simply process with a infusion reaction, which means Gibson reaction, to fuse the two ends together and to obtain our plasmid with designated mutation. These two reactions both is targeting on a single nucleotide mutation and is both processed with PCR, but the only difference is that the substrate we are targeting is different. We will cover how to design such primer for the site-directed mutagenesis next week, and now you might know how to choose the correct approach to manipulate your DNA sequence, coding sequence for your protein. Thank you.